Good day. I am Zaki Suficiencia, and on behalf of my co-authors Jed Allen Hernandez and Dr. Maria Carmen Ablan-Lagman, I will be discussing our study entitled TLR4 and IFNG are candidate genes for the study of variation in Philippine chicken. The chicken industry is an important component of the economy, providing several products such as eggs and meat. The United States and Brazil are two of the major chicken product exporters. China is also a major chicken product exporter, although lesser in scale compared to the previously mentioned countries. In the Philippines, the chicken industry is also an important part of the economy. They provide a source of livelihood through the selling of meat and eggs, which can also be harvested for personal consumption. As of 2020, the Philippines produced 1.81 million metric tons of dressed chickens and 605.79 thousand metric tons of eggs. The industry, however, has always faced challenges with disease. This has generally increased due to the practices of overcrowding and the developing risk of antibiotic resistance. As such, there has been a shift in focus to genetics-based breeding programs to combat this. It has been established that genetic variation and disease resistance or tolerance are correlated with each other. Depending on the genetic profile of an animal, in this case the chicken, they may either be more susceptible or resistant to certain types of diseases. Given the importance of genetic profile, it is important to look into novel genetic variants which confer attractive traits. One such source are the indigenous breeds of livestock animals. Their adaptation to the local environment results in the selection of desirable traits, such as resistance to common endemic diseases and tolerance to extreme temperatures. These indigenous populations can be thought of as reservoirs of genetic resources which can be used to improve commercial chickens. The protection of these reservoirs is important given that commercial chickens have extremely low genetic diversity worldwide. In order to utilize these reservoirs effectively, there must first be sufficient knowledge about their genetic resources. This is particularly lacking in Philippine chickens, which have had very little genetic characterization done. On top of this, the number of native chickens in the Philippines has steadily dwindled, with farmers preferring to raise faster growing commercial chickens. While commercial chickens have the advantages of faster growth and size, they have had problems with decreasing genetic diversity in their populations, making them more vulnerable to epidemics. Investigating the genetic resources of native Philippine chickens may provide a means of increasing the genetic diversity of commercial chickens through breeding programs, with special focus to the improvement of their immunity. The investigation of genetic resources will focus on genes that play a role in immunity or disease resistance. The selection of such genes can be done with the aid of sequence repositories and gene ontology analysis. In line with this, the study reviewed the current information on the genes related to immune response in Gallus gallus to determine those that are shared across different diseases, retrieved the sequences for the genes that meet the criteria of numbers and quality, and evaluated their variation in preparation for a comparison with the native Philippine chicken. This study provides future researchers with a general direction for the study of variation and breeding for disease resistance in Philippine chicken genetics. First, we did a literature search to collect the genes implicated in disease and which diseases we will cover depending on the availability of literature. We collected our literature from Google Scholar and PubMed using consistent search terms. These genes were then recorded and listed down. For gene analysis, we performed a functional enrichment analysis using gProfiler to develop gene ontologies per disease, which characterize the genes involved in each of them. The genes were then scored based on how many diseases they were implicated in using Microsoft Excel, from which a list of the top genes implicated in diseases were developed. From the top 6 genes, TLR4 and IFNG were considered for haplotype analysis due to having a substantial number of published sequences for Gallus gallus. We retrieved Gallus gallus sequences of both genes from GenBank. MegaX was used to trim and align the sequences. The DNA-SP application was used to detect haplotypes, while the SNP sites application was used to tabulate SNPs. A modified R script from the Parslan et al. was used to create visual representations of the haplotype data and networks. More than half of diseases considered are bacterial in nature, followed by viral, and a single protozoan. Most of these diseases have been reported in the Philippines, with the exception of lymphoid leukosis and necrotic enteritis. However, these are still included in the study due to the possibility of future outbreaks. An observation we found was that the respiratory, lymphatic, and gastrointestinal systems are the systems most commonly affected by these diseases. From the 73 papers collected, 428 genes were considered after the cleanup of data. The abundance of genes found indicate the potential variety of response mechanisms and complexity of gene expression in chicken diseases. In order to determine the nature of these genes, gene ontologies for each disease were developed. The graph shows the number of GOCC, BP, and MF terms for the first 8 diseases. This next graph shows the GO terms for the remaining 7 diseases. The abundance of genes detected in the literature review indicate the potential variety in response mechanisms to a pathogen and the complexity of the gene expression processes that are involved with disease in chickens. Majority of the genes identified in the analysis were involved in the immune system. In regards to the abundance of terms, GOBP consistently had the highest number of terms, followed by GOMF, then GOCC. This trend may be due to 2 reasons. The genes implicated in the disease may be considerably unique from each other, thereby reducing the number of shared terms between them. On the other hand, it may be that the terms the genes share truly are limited in number due to their similarity. Of the 428 genes, only 55 were found to be common in at least 2 diseases. Only 6 genes were involved in 5 or more diseases. From the data, we could see that there is more uniqueness in terms of response than commonality. Targeting genes involved in multiple diseases can be beneficial by improving overall resistance to disease. While the current paradigm for chicken breeding programs is a focus on economic traits like meat quality and size, these genes can be utilized in future breeding programs centered on immunity given the threat of possible epidemics. In Philippine native chickens, there must then be more in-depth investigations in the genetic variation of these immune response genes. In terms of breeding programs involving native Philippine chickens, on one hand, breeding could be done with multiple genes conferring attractive disease response traits for a maximal increase in disease resistance. On the other hand, breeding could also be done with a focus to a single gene with an attractive disease response trait, although this would have a lower impact on overall disease resistance improvement. This table shows the top scoring genes in the study and the number of available sequences online. The 3 highest score genes, IL-8L2, IL-6, and IL-10, only had a few available sequences. 3 genes had a sizable number of available sequences, namely IL-1B, IFNG, and TLR4. Only IFNG and TLR4 were chosen for further analysis because almost all sequences of IL-1B came from a single publication. Implications from this further analysis will then be put into the context of Philippine native chickens. After retrieving the sequences, a haplotype analysis was done. TLR4 has 94 haplotypes, with 30 SNPs spread across the gene having a length of 2,532 base pairs. Among the haplotypes, only 4 haplotypes had a significant bootstrap value between 50 to 85%. Generally, branches with bootstrap values of more than 70% can be regarded with high confidence as being true in nature. The low bootstrap values could be indicative of silent mutations which do not affect the protein produced. Hence, it is not significant, but further tests should be done to confirm this. The haplotype network visualizes the relationship among the haplotypes. The 94 circles represent the 94 haplotypes detected within the sequences used in the study. Among the haplotypes, haplotype 86 has the most individuals, having 14 sequences, and represents the origin of the haplotypes. There is an evident linear progression in haplotypes of TLR4, which implies a fast mutation rate. Evolutionary rates may be associated with its function in pathogen recognition. TLR4 in chickens recognizes lipopolysaccharides of gram-negative bacteria. Lipopolysaccharides are an example of PAMPs recognized by PRRs such as TLR4. PAMPs associated with TLRs tend to experience selective sweeps, which TLRs must adapt to accordingly. The IFNG neighbor joining tree indicates that there are 7 haplotypes present among the sequences. All branches have low bootstrap percentages, with none having a significant bootstrap value. This indicates that there is low confidence for the relationships between haplotypes. This is the haplotype network of Gallus-Gallus IFNG. The Gallus-Gallus IFNG gene had 7 SNPs and 7 haplotypes. The majority of sequences belong to the central haplotype, which is haplotype 2. Haplotype 2 is the probable origin of the remaining 6 haplotypes, since all branch out from it. The radiated branching character in the haplotype network of IFNG contrasts with the linear one of TLR4. It indicates lower mutation rates or less tolerance to mutation. It may be slower in IFNG due to its many essential functions in the immune system. It links the response of the innate and adaptive immune system by promoting MHC2 expression and regulates the function of macrophages, natural killer cells, and B cells. Given the many functions that IFNG has, even small mutations in its sequence could reduce its effectivity in these activities. It will be interesting to determine how the Philippine chicken genes would figure in the patterns demonstrated by the samples already studied. Backyard-raised native chickens in the Philippines often scavenge for food and water. Feeds are broadcasted into the ground, which gives them more exposure to potential pathogens. This may result in the selection of more disease-resistant phenotypes controlled by these 2 genes. In conclusion, 73 unique papers were reviewed on genes implicated in disease response across 15 selected diseases of global economic importance. 428 unique genes were found, and 55 of these were implicated in 2 or more diseases. This implied the diversity of pathogen processes and the need for the enhancement of an array of genes and pathways. Functional enrichment analysis provided insights into the shared biological terms of the genes, helping in their characterization. The majority of the terms were involved in the immune system process. Of the top 6 genes, TLR4 and IFNG were chosen for haplotype analysis. Haplotype analysis showed the differing mutation rates between TLR4 and IFNG. TLR4 experiences higher rates due to its function in pattern recognition. For recommendations, it is suggested to test the remaining genes in the top 6 list for their variation. This would require an extensive collection of samples and sequences, as online databases lack this, with the exception of IL-1B. In addition, it is recommended that studies be done per pathogen type to garner more specific insights to genes involved in their disease response. Finally, lab samples from Philippine native chickens should also be tested for any variation in these genes. That is all. Thank you for listening.